DNA loading dye is an essential reagent used in molecular biology during gel electrophoresis. Its primary role is to prepare DNA samples for easy and accurate loading into the wells of an agarose gel. The dye contains two important components, tracking dyes and a dense substance like glycerol or fickle. The dense component ensures that the DNA sample sinks directly to the bottom of the well, rather than floating away in the buffer. The tracking dyes, usually visible as blue or purple bands, help us monitor the progress of electrophoresis as they migrate through the gel at predictable rates. To prepare the sample, a small amount of loading dye is added directly to the DNA solution. This mixture is then gently mixed to ensure uniform distribution without creating bubbles. Once ready, the sample is carefully loaded into the wells of the gel using a micropipette. During electrophoresis, the electric current causes DNA fragments to migrate through the gel toward the positive electrode, and the tracking dyes move along as visual markers. These dyes do not bind to DNA, but they indicate how far the run has progressed. After the electrophoresis is complete, in summary, DNA loading dye plays a crucial role in ensuring samples are properly loaded and that the electrophoresis process can be visually tracked, making it a key step in successful DNA analysis.